Good morning, and welcome to Medicine Grand Rounds. It's really my pleasure to introduce Matthew Merrins, our Grand Round speaker. Matt received his BA in Biochemistry and Biology with honors from Oberlin College, and then went to the University of Michigan, where he received a PhD in Physiology. He then undertook a postdoctoral fellowship in Michigan and was a research investigator at the University of Michigan until he was recruited by Vince Crines to join the Division of Endocrinology, Metabolism, and Diabetes as an assistant professor. He's also an assistant professor in the Department of Biomolecular Chemistry and is a research scientist here at the William S. Middleton Memorial Veterans Hospital. He's also involved throughout the university, involved the integrated program in biochemistry, molecular biophysics graduate program, integrated graduate program in nutritional sciences. He's involved in the Carbone Cancer Center cell signaling program, the endocrinology and reproduction physiology graduate program, and the cell and molecular biology graduate program. He's already published 15 peer-reviewed articles, 6 of them since arriving in Wisconsin, and has presented 28 nationally presented abstracts. He's received significant grant support, currently sponsored by the NIH for a KO1 and an R21 grant, and he's also PI on an American Diabetes Association Basic Science Award. He's been an active teacher over the years. At Michigan, actually received 4 teaching awards, and he is on 7 thesis committees here at the University of Wisconsin, 2 acting as chair. He's a very good citizen locally and nationally. He's chair of the VA Research Grant Review Committee here in Wisconsin, and he is now serving as a senator for the University Faculty Senate representing our department. He's an ad hoc journal reviewer for 11 journals. Along the way has received a number of grants and awards, including most recently the American Diabetes Association Innovative Basic Science Award. He's given a number of national lectures already as well, most recently at the American Diabetes Association Scientific Sessions and the Department of Physiology at Yale. Today he's going to share with us some of his own research and give perspective for Grand Rounds in his talk entitled "The Role Reverse in Basic Science Lab, Aged Mice Proposed to Stop Diabetes in Human." Please join me in welcoming Dr. Matthew Merrins.
Thanks, Rick. Well, good morning, everyone. It's a pleasure to be speaking to you this morning. So I'm going to focus my talk this morning on the topic of diabetes, and I want to start with some quick facts about diabetes here in Wisconsin. So diabetes is not among the leading causes of death. It's only 3% of deaths that it accounts for in Wisconsin, but it's predicted to be one of the most socially and economically destabilizing factors of our lifetime. And there's a few reasons for this. So one thing is that currently 1 in 10 adults in Wisconsin has diabetes, and 25% of the people with diabetes just don't know that they've got it. One of the problems that we're facing is that in Wisconsin, 30% of the adult population is obese and at risk for type 2 diabetes, which, as most of you know, is the majority of diabetes. So the sum total of all of these effects is $6.15 billion in annual health care costs to the state residents of Wisconsin. And $4 billion of that is just direct costs alone. There's another $2 billion that's hard to account for, that's lost productivity. So these numbers are pretty staggering, but what's worse is what's about to happen. So one of the central problems is that the median age of the US population has been rising for 30 years and is projected to continue to rise. And age is a major risk factor for type 2 diabetes. So diabetes prevalence increases with age, and 1 in 4 seniors currently have diabetes. Another problem, which I think everyone in this room knows about, is that obesity prevalence has been rising for a number of years. And it's projected to reach 50% by the year 2030. So the combined effects of these two problems are that children born today have a lifetime risk of developing diabetes that's estimated to be 40 to 50%, which is a pretty huge number. So my laboratory is focused on age as a risk factor for diabetes, alone or in combination with obesity. And that's really where I'm going to focus my talk today. So today's talk is really going to be about my laboratory's approach to this problem. There are a lot of approaches being taken in the Department of Medicine. And the one I'm going to tell you today, as a basic science researcher, we sort of have two main types of studies that we do in the laboratory. One type is where we have a particular gene that we think is a risk factor for diabetes, and we knock it out, and we find out its cellular function. But that's not the kind of study I'm going to talk about today. I'm going to talk about an untargeted or unbiased study where we do a discovery-driven search for drug targets using the physiological pressure of aging. And while I'm doing this, I want to highlight the tools that are available to us here at Wisconsin. And to stay sort of at the state-of-the-art place, what this is going to involve is some biophysics. And so buckle your seatbelts. But I've tried to work really hard to make this presentation accessible for everybody in the room because I know we have a really diverse audience. So those are the implicit goals, but here are the explicit goals of my presentation today. So in the first half of my talk, I want to introduce to you a discrepancy between glycemic control between aging humans and mice. I want you to understand the physiologic underpinnings of this discrepancy. It happens to be an ion channel in the beta cell called the ATP-sensitive potassium channel, or KTP for short. Now, because this channel is druggable, I want to discuss the practical implications of targeting KTP. And finally, I want you to understand how obesity impacts the aging beta cell, because that's a separate challenge from the effects of age alone. So my story starts here with the pancreatic beta cell, which releases the hormone insulin in response to blood sugar. Now, beta cells are part of a larger structure which is called the pancreatic islet. And the islet contains a number. So AV is going to have some problems today, so just to warn you all, I'm just going to keep plowing ahead. Vince tells me that, or not Vince, Clint tells me that the blue screen of death will disappear. So these pancreatic islets, they're filled with hormone-releasing cells, including the alpha cell, which releases the hormone glucagon, and glucagon opposes insulin's action. And so together, these hormones that are released from the islet regulate blood sugar within a very narrow range, about 3 to 5 millimolar. So the beta cell does this by responding to elevations in blood glucose after a meal with a proportional release of insulin. Insulin then, by acting at its target tissues, promotes nutrient uptake, and that negative feedback lowers blood glucose back down to its set point. So the failure of this process from the beta cell is the primary defect in diabetes. We've known this for decades, and we still do not have an answer to this. So most of you in the room will have had some exposure to this homeostatic feedback loop, but I wonder how many of you know that insulin release oscillates. It's pulsatile. So insulin secretion oscillates in humans. It oscillates in non-human primates, in dogs, in rats, and mice. And these oscillations have a period of several minutes, and they're absolutely critical for insulin's action at the liver, especially. So you can also see that these pulses are lost in type 2 diabetics. And Robert Turner's lab, who did this work in the late 70s and early 80s, also showed that the oscillations are lost in the obese but non-diabetic relatives of these individuals. Now I want you to remember this slide because in the middle of my talk, I'm going to come back to the pulsatility of insulin secretion, which is generated by the beta cell itself. And I'm going to show you how we can analyze the shape of the oscillations to pinpoint the defects in the beta cell secretory pathways that are deficient in a disease state. So that's the power of our approach. The final point of my introduction is that glucose triggers insulin secretion through two key pathways, which can be modeled by a light switch that you might find in your dining room. So there's an on-off switch, or a trigger, in series with a dimmer switch, or an amplifier. So the beta cell works precisely this way. In the triggering pathway, glucose that's taken up by the beta cell is metabolized through the processes of glycolysis and the mitochondria. And then the mitochondria, as the powerhouse of the cell, sorry about that. The mitochondria, as the powerhouse of the cell, raises the cellular level of ATP and lowers the cellular level of ADP. And these two messengers travel to the plasma membrane where they inhibit an ATP-sensitive potassium channel. So that inhibition triggers calcium activation, and then calcium is the direct trigger for insulin release. So if you only remember one thing from that entire biochemical pathway, remember this channel, the KATP channel. It has a special property. The special property is that it is sensitive to metabolism. It is the metabolic sensor for the plasma membrane. And the number of KATP channels in the plasma membrane determines how much glucose it takes in the blood to turn insulin secretion on or off. The second major pathway is the amplifying pathway. So the amplifier simply determines the precise number of insulin granules that are released every time the switch is turned on. All right, so the key points to take home from my introduction. First point was that pancreatic beta cells are nutrient sensors for the organism. They use metabolism for signaling, not just their own energetic needs. So metabolism is a signaling pathway. So if there are defects in diabetic beta cells, metabolism is a great place to look. My second point was that insulin secretion is pulsatile. These pulses are lost in type 2 diabetics, and these pulses are essential for insulin action. So this is a pretty big problem for people who are producing insulin pumps. Third, glucose-stimulated insulin secretion resembles this toggle dimmer switch. So the toggle switch determines when the pulses of insulin are on or off, and the dimmer determines how much insulin is released during each pulse. And here's the focus of my talk today. 2 risk factors for type 2 diabetes in particular. Age alone or in combination with obesity. So I'm going to talk about age in the first half of my talk, and age and obesity in the second half of my talk. And as a field, as an islet biology field, and as an aging field, we still do not know how aging affects beta cell function. Okay, so to get us started, I want to talk about some data that I collected in collaboration. Actually, I didn't collect it. Michelle Kimple collected it. So we collected this data in collaboration with the Kimple lab. And Michelle, over the course of her studies, measured insulin secretion from 31 human islet donors. So these are islets that have been donated for research. They arrive in the mail, and then we can study them. So these islet donors were aged 15 to 64 years of age. And I asked Michelle to take the data that she had already collected and plot glucose-stimulated insulin secretion, which is shown in red, 17 millimolar glucose, with age. And you can see from the regression line that there is a sharp decline in glucose-stimulated insulin secretion with age. But that at least in our study, does not also apply to basal insulin secretion. So these results are not due to defects in insulin synthesis. So the insulin content of those islets has no trend with age. And they're also not due to any randomness in the BMI of the donors that we received in the mail. So this is really an age-dependent effect on insulin secretion. So I want to contrast this with what we found in mice. And here comes the big surprise. So insulin secretion increases with age in mice. And since the 1980s, people have not believed this to be the case. But I want to show you our data. So mice that are 6 months old and 24 months old were obtained from the NIA, the National Institute of Aging. And they keep a colony of old mice for researchers, which they send to us as part of our grant, free of charge. And then that saves us from having to wait 2 years to obtain these animals, so we can get our studies going a little more quickly. So in collaboration with Dudley Lamming's lab, we measured glucose tolerance in these animals as a measure of organismal glycemia. So how this works is we fast the mice overnight. Their fasting blood glucoses are under 100. And then we inject them with 1 gram per kilogram glucose. Blood glucose goes up, and then insulin and other factors bring it back down. And you can see there's absolutely no difference in glucose tolerance between the young and aged mice. And this is surprising, because humans get glucose intolerant with age. So when we measured insulin secretion in vivo, we injected glucose, and 15 minutes later, we saw that more insulin was released in the aged animals than in the young animals. And when we isolated the islets and measured insulin secretion ex vivo, or in a tissue culture dish, what we found is that either at low glucose or at stimulatory levels of glucose, here we're using a hammer, 17 millimolar glucose, we find that insulin secretion is again increased in the aged mice. Now, my group is not the only one to challenge this idea that insulin secretion declines with age in a mouse. There's other corroborating evidence just from the last year alone. So Klaus Kessner's lab published in Cell Metabolism, Itai Ben-Porath's lab published in Nature Medicine. They say exactly the same thing that we say, which is insulin secretion is actually enhanced in aged animals. Although one study, I have to tell you, in full disclosure, found no change. But all of the studies agree that the dogma, which has been in place since the early 1980s, does not appear to hold up. And there are some flaws with those measurements, which I can go into. So this brings me to the central question of my talk. So what underlies the difference in the beta cell secretory phenotype between the aging humans and mice? And you can kind of see the glass as half full or half empty. So on the one hand, you might say, all right, well, this means that these mice are terrible models for human aging. So, okay, so that might be a potential downside. The upshot is what if we can find the mechanism of compensation in the mice and leverage it for therapeutic gain in humans? That's the upshot. Okay, so that's what we're going to try and do. So this is a story about making lemonade. All right, so what's required to do this? What we really need a systematic analysis of the beta cell secretory pathway that I showed you. And where to start in this pathway? Well, one compelling hypothesis could be that there are species differences in mitochondrial function. So mitochondrial failure is considered by the aging field to be a hallmark of aging and cell senescence, but that has not been studied in pancreatic beta cells. So another reason this hypothesis is pretty compelling is that mitochondria are enmeshed in the beta cell's glucose sensing apparatus. Remember I told you that beta cells use metabolism for signaling, not just for their own daily energetic housekeeping needs. They use it to sense glucose and trigger a proportional release of insulin. So we took this opportunity to examine the mitochondria and islets to develop some new methodology for assessing mitochondrial function, specifically in this cell type. And so in this talk, I'm going to focus on the work of my graduate students who are here in the audience. So Trilly Gregg, who's a biophysics graduate student, and Sophie Sadeo, who's an IPIB graduate student. So what we did in our last paper, which was published in Diabetes about 2 weeks ago now, is we introduced the islet field to NADH fluorescence lifetime imaging. And this is something that some of you in the audience, I see Roz Anderson is here, know about already. But this is a brand new approach to the islet field, and I want to tell you how it works. So this technique takes advantage of the endogenous fluorescence of NADH that's present in the cell. Now, as you can see from this image of the pancreatic islet, most of that fluorescence is mitochondrial. So all of those little dots and worms that you see in white here, those are the mitochondria. And the dimmer spots are the nuclei and the cytosol. Now, since the 1960s, 1960s, sorry. Britton Chance did this work in the 60s, not the 80s. So since the 1960s, people have used NADH fluorescence as a method to evaluate metabolism in living cells and in real time. And you can see that when we take islets and we raise glucose from 2 to 20 millimolar, there's a strong increase in the fluorescence of NADH. And that fluorescence increase is abolished when we add a mitochondrial poison, a CCP. Now, an important thing to note is that the NADH intensity measurements that you see here do not reflect the concentration of NADH levels. So most of this fluorescence actually comes from NADH binding to proteins. So in parallel with these binding events to proteins, there is a change in what's called the NADH fluorescence lifetime. Now, what is that? This is where we get to the biophysics. So the lifetime is the time a fluorophore spends in the excited state before emitting a photon. And you don't need to care about that. You just care that we can measure it. It's very short. It's only a few hundred picoseconds. So if we can measure the NADH lifetime directly, it can tell us which NADH binding proteins the NADH is bound to in a living cell, because there is a unique NADH lifetime for every single protein interaction. So what an NADH lifetime image is it's a spatial map of all of the redox-dependent pathways, or NADH-dependent redox pathways, in a living cell. And the goal here is to use this technology to compare the healthy cells to the diseased cells and see what's different. Now, with the current state of things, our current technology, we can't resolve all 379 NADH-dependent coenzymes with this technique. That's just simply not possible. We're a number of years off. But we can resolve one that we're very, very keen on in the mitochondria. And that's mitochondrial NADH production by the TCA cycle. And so just to remind you, if it's been a while since college biochemistry, so this is a schematic of the mitochondrial electron transport chain. And what the cell is doing in the mitochondria is it's using NADH and consuming it at 3 different complexes in this chain, complex 1, 3, and 4, to pump protons into the inner membrane space. Now this process consumes oxygen, and then at the end of this chain, at this complex 5, ATP is produced. And remember that in the beta cell, the ATP goes on to become a trigger for insulin release. So what we did with this technique was we measured the NADH lifetime in islets that we received from these human donors in the mail. So what you're seeing here is we took every pixel of these images and we plotted them as a histogram. So this is a graphical representation of every NADH-dependent pathway in these beta cells, where the vertical axis is simply a measure of how often a particular lifetime interaction occurs in the cell. But the key feature here of these plots is that on the horizontal axis, electron transport chain function increases as you move to the right. And what you can see from our plots of histograms of aged islets and young islets is that the aged islets lag behind the young donors. So what we did was we plotted the histogram peaks versus age for a number of these donors. And you can really see from the regression line, with actually just a few donors, the regression statistics are quite strong. You see a decline in mitochondrial function in the human islets with age. Okay, that was all of the hard biophysics. I swear it gets easier from here. All right, so what we did is we repeated this study in the mice, where insulin secretion is enhanced. And we asked, well, does mitochondrial function also fail here? And it does. So again, the aged islets are lagging behind the young. And we made these measurements at 2 different levels of glucose, and you can see the effect is on electron transport chain function is quite strong. This pathway is very depressed. And just so you know that what I'm saying is biochemically sound, because we've introduced a new technique to the islet field, we did things the old-fashioned way also. So we used the classic biochemical measurement of oxygen consumption rate. Actually, thank you, Department of Medicine, CapEx. We used an Ouroboros to do this. And we found a 55% decline in mitochondrial respiration in the aged islets. So this is really surprising because the central pathway that triggers insulin secretion has a hallmark of aging defects, which is mitochondrial deficiency. So this rules out our first hypothesis. There are basically no gross differences in mitochondrial function between these two species that I'm showing you. So now what? So here's our mitochondrial decline. Here's our increased insulin secretion. There's got to be something in between the mitochondria as an organelle and insulin secretion that compensates to give us this phenotype of enhanced insulin secretion with glucose. So we decided to look at intracellular calcium. So calcium, remember, is the direct trigger for insulin release. So remember I said I would come back to pulsatility in my talk. So here we are again. So this is a movie which has 15 pancreatic islets from mice, and they contain a calcium dye, and you can see that there are oscillations in calcium. Now these pulses of calcium are the pulses that drive pulsatile insulin secretion. And I want to tell you how we can use the shape of these oscillations to understand where the defect is in the biochemical pathway that I'm showing you here on the left. So let's do that. So to do that, I want to go back to our model beta cell, which is this light switch, and the triggering and the amplifying pathways. And I need you to understand the specific behavior of this on-off switch, this trigger. It's not just always on or always off. There's a nonlinear relationship with glucose. So if we're measuring intracellular calcium, which is a good proxy for insulin secretion, and glucose levels are low, they're below 7 millimolar, so this is 5 millimolar, calcium is low and stable. At intermediate levels of glucose, 7 millimolar and 10 millimolar, the islets are on. The insulin secretion is on with each one of these pulses. The islets are oscillating. And then at really high levels of glucose, which are only seen in pathophysiology, the islets never turn off and calcium is high and stable. So the important point here is that this set point for turning insulin secretion on and off, this exact trigger or switch, is set by the precise number of ATP-sensitive potassium channels that are present in the plasma membrane. The second important point about this diagram is that within the oscillatory regime, the pulse width increases with glucose. That's the width of these oscillations. But what doesn't change is the frequency. So if there are anyone who studies neuroscience in the room, beta cells do not encode information like neurons. So a neuron, let's say that my brain needs to know if I'm holding a heavy book in my left hand or a lighter book. When I'm holding the heavy book, the stretch-gated ion channels in my hand fire more frequently to my brain to tell it that I'm holding a heavy book. Beta cells don't encode information like this. They encode it in the fraction of time that they're active. And this is the analysis that can help us understand where the defect is. So from what I just told you, you can look at the shapes of these calcium oscillations collected from young islets on the left in black and aged islets in red and see that the peak width of each oscillation is broader. And that's quantified on this graph. So what we find that calcium is actually enhanced despite the fact that the mitochondria are deficient. So our new hypothesis is that a change in the number of KTP channels underlies this basic fundamental compensation for aging in mice only. So this turned out to be the case. And so let me prove this to you. So what we did is we measured the number of KTP channels present in the beta cell directly. So we can do this with a technique called patch clamp electrophysiology. And so the image on the left shows you a pancreatic islet. I took its picture with our microscope. And that blurry bit that you see is our electrode, which we've hooked up to a beta cell on the surface of that islet. The cartoon shows you how the experiment works. So inside the plasma membrane of the beta cell, there are a number of ATP-sensitive potassium channels. And using the electrode, we can measure the flow of current through those channels in the presence of a certain amount of glucose. And then again, after we've opened up every single channel with a drug, in this case we're using a KATP channel opener called diazoxide. And what we found is, in the age cells, there's reduced potassium channel current, which means there's fewer channels in the plasma membrane. And we can quantify this on this graph right here. So we see this strong decline in ATP-sensitive potassium channels. And what we think is this is basically a fundamental compensation in the aging mouse beta cell. This compensation isn't just present in the context of age. You can find it in other contexts, and I don't have time to go into them now. But the take-home point of our entire story is that we started with this phenotype of enhanced insulin secretion and reduced mitochondrial function, and we were able to find the site in the biochemical pathway that underlies this change. So to summarize what I've told you thus far, we've started with an aging organism, or two aging organisms, humans and mice. And I talked about a difference in glycemic control that occurs with age between these two species. And the mice uniquely are able to respond to age with enhanced insulin secretion. And we traced this to the level of the single cell, to the beta cell. We did a pathway analysis of the beta cells, and finally we ended up at a single molecule, the ATP-sensitive potassium channel, which is a druggable target. Now those of you who are in endocrinology are going, wait a second, I knew that already. We've heard of this channel. We've been using these drugs called sulfonylureas since 1942. So this is the downside of these discovery-based studies. I cannot predict what the compensatory mechanism is going to be. All I can tell you is that in an aged beta cell, no one looked. And we found something that we already knew about, and I'm really sorry about that. So the story of these drugs, though, if you don't know, it's really kind of interesting. So there are ATP-sensitive potassium channel blockers, like tolbutamide. That was in the first generation. Second generation included things like glyburide, glibenclamide. And these are really mostly used to treat neonatal diabetes. But I happen to know a little bit more about this because the three guys who really discovered the efficacy of these for neonatal diabetes worked at the University of Michigan, where I trained, and at the University of Chicago. And what they had been doing was they had been treating patients with neonatal diabetes, and these are parents who are staying up all night injecting insulin into their kids for years. And what they did was they started mapping out the genetic underpinnings of this problem. And this is a small subset of diabetes that's monogenetic. But there are defects in this ATP-sensitive potassium channel, mutations that cause this neonatal diabetes. And when they gave the kids low-dose sulfonylureas, they basically went home fine. So it's a really nice story, and it already happened. All right. So that gets me to everything that I've been sweeping under the rug for the last half an hour. So some of you should be waving a flag and saying, wait a second. There is an important confounder in all of this. And that is, take a look at the BMI of our human islet donors. 50% of them are clinically obese. Now, I'm comparing humans, which got fed a Western diet for most of their lives. These are not healthy donors, the ones we received in the mail. These are the people that have died prematurely, for the most part. So we've been feeding these mice a low-fat diet mouse chow. So how is that a fair comparison? And the answer is it's not. Now, we don't have enough data for me to show you what the phenotype of the normal aged human donors is relative to the human donors with a really high BMI. You can see we've only got several samples here. The lab's only been open for a couple more years. Hopefully I can update you in this in a couple more years. But what we could do immediately was we could correct for this confounder by putting mice on a Western diet. So in collaboration with Dudley Laming's lab, what we did is we took our two groups of mice, our young mice aged 6 months and our old mice aged 22 months, and we put them both on a Western diet or a chow diet for 8 weeks. And at the end of this, we measured organismal glycemia with a glucose tolerance test. And we found something that was pretty surprising. What we found is that the young mice responded to the Western diet by getting glucose intolerant, which is what we expected. But the aged mice were bulletproof to this diet. Put them on a Western diet, they don't get glucose intolerant. There's something special about the aged mice. Now, that brings up another key question, which is going to be the focus of the remainder of my talk, which is what allows the aged mice to tolerate overnutrition. Now, there's a classic way of asking this question in science and a not-so-classic way. The classic way is to say, all right, well, we've got young mice on a Western diet. Let's compare those to aged mice on a Western diet. Now, all of the aging researchers in the room are going to think and know already that this is a terrible comparison because so many things change with cell senescence. Over the 2-year lifespan of this mice, these mice lived around 28 months of age, there are myriad changes in the epigenome, in the transcriptome, that I can't control for in this experiment. So the real experiment, which almost nobody does, is to only study this one group, this aged mice on a Western diet. And so we took a bet. We bet that the resolution of the tools that we have in the laboratory is so good that we could tell you where it is in the beta cell pathways that underlies compensation by comparing the mice that compensate for Western diet to the ones that don't. In most studies, that's a single treatment group. We're going to delve into the treatment group and look at individuals. So this analysis is actually very powerful. So what we did is we had to find a way to divide up the mice into groups. And we decided to do that by weight and by glucose tolerance. So it turned out that the mice that weighed 40 grams or less after we fed them the Western diet had normal glucose tolerance. That's the curve in black. So we call these the NGT mice, and that's how I'm going to refer to them. To contrast that, the mice that weighed more than 40 grams at the end of this treatment had impaired glucose tolerance. We're going to call those the IGT mice. And when we looked at insulin secretion in the IGT mice, what we found is that it was depressed. So the IGT mice release less insulin than those that maintain glucose tolerance. So we started our biochemical pathway analysis again in this cohort of animals. And once again, we started with the mitochondria. Although this time we measured a different feature of the mitochondrial electron transport chain than we did in the first half of the talk. We measured the mitochondrial membrane potential. So the membrane potential occurs because the respiratory complexes in the chain are pumping lots and lots of protons out of the mitochondrial matrix and into the inner membrane space. And protons are charged, so when they do this, this leaves a negative potential on the inside of the membrane and a positive potential on the outside of the membrane. And that potential is later used in the last step of the electron transport chain because when the protons come running down their concentration gradient, they run down this electrical gradient, and that helps generate a force for ATP production. So that's just basic mitochondrial biology. So this applied to the islet. What we did is we took a dye that's sensitive to the membrane potential, and we took our NGT islets, which were isolated from NGT mice, and our IGT islets, and we raised glucose from 2 to 20 millimolar, and we measured how much the mitochondrial membrane potential would become activated. And we standardized all of these measurements with potassium cyanide. This is basically just a control for the islet size. Different islets have different sizes. You have to control for it. So this gives us a basement to normalize to. But the main point of this result is that the mitochondria in the mice with normal glucose tolerance were more active by a lot. So what this means is that there's a mitochondrial effect right off the bat. So how about downstream of the mitochondria? So the next measurement we did was one where we measured the triggering pathway. And remember, this is the on-off switch that carries the oscillations. So what I'm showing you here are the first triple simultaneous measurements of 3 parameters in a living cell. One parameter is mitochondrial NADH production. Another parameter is the downstream change in the ATP-ADP ratio. And the final parameter is calcium. So you can see that there are oscillations in each of these parameters. And this was done by a technician in my lab, Brian Schmidt. This has not been possible before. We're going to publish this hopefully this fall. And, well, thank you, Startup Package, because this has not been previously possible. So you can see that the shapes of the oscillations are really different. And I don't have time to go into the biochemical underpinnings of those differences. But what I want you to notice from these traces of young islets stimulated with glucose, NGT islets stimulated with glucose, and IGT islets, is that when we choose a glucose level that's right near the threshold for turning the beta cells on and off, what we find is the mice with normal glucose tolerance have insulin secretion that's turned on, and the mice with impaired glucose tolerance have insulin secretion that's turned off. So this compensation, quantitatively, means that in NGT islets, 60-some% of them are oscillating at intermediate levels of glucose, but only 18% of them in the IGT islets. So this compensation could be due to changes in the ATP-sensitive potassium channel, which you learned about in sort of the middle part of my talk. We don't know. We haven't tested that yet. But what I want to focus on in this last bit is this other pathway, the amplifying pathway. And I haven't said much about this yet. So remember that the mitochondria in the beta cells from mice with normal glucose tolerance are more active. And the mitochondria control not only this triggering pathway, but they also generate second messengers which raise the level of insulin secretion. This is the site of a lot of drug targets inside the amplifying pathway, and there's probably even more. As a beta cell field, I can't even tell you all of the coupling factors that this red arrow represents. There's a lot of them, and we just simply haven't gotten there. So to measure this pathway, though, we can measure it directly and in isolation. So how we do this is shown by this cartoon. So what we're going to do is measure what we call KATP and independent secretion, which is just the amplifying pathway. What we do is, once again, we use the patch clamp technique. We hook an electrode up to a single beta cell on the surface of the islet, and we give a pulse of electricity which stimulates the calcium channels directly in these cells. And calcium is the direct trigger for insulin release. So when calcium levels rise, insulin vesicles fuse with the plasma membrane. Now, how the technique works is that we can measure the cell's capacitance. So the capacitance is an electrical feature. But what this really translates to is the cell surface area. And every time we add an insulin vesicle from the pool that are ready to be released to the plasma membrane, it increases the surface area, and we simply count the number of vesicles that are added. So it turns out that it takes about 2 femtofarads of capacitance change for every granule. So in these beta cells from the mice with normal glucose tolerance, when we stimulate them 10 different times to activate their calcium channels, they release about 320 femtofarads of capacitance, about half that in insulin granules, so about 150, 160 insulin granules. Look what happens with the mice with impaired glucose tolerance. This amplifying pathway has really been altered in these mice. So, to get you to the conclusions of my talk, what we've done here is some discovery-driven studies using aging and obesity as the pressures that we put on the organism to find and see if we can find drug targets. And what I showed you today was that the impact alone on organismal glycemia has to do with the beta cell, and it has to do with this light switch, this triggering pathway. And that was the ATP-sensitive potassium channel effect that I showed you. Age and obesity have separate and distinct effects from that of age alone. They affect how much insulin release we get every time there's a pulse of calcium. The second conclusion is more of a thought experiment. I'm not sure if it can be a conclusion or not. So I think it's possible that what these studies say is that beta cells are a primary driver of glycemia or glycemic control in aged animals. Now, glycemic control is a really complex parameter. So I don't think there's any way as a basic scientist I could prove this to you. But I have a thought experiment for you that supports my argument. Suppose, for a minute, that all of these effects of age and obesity to rescue organismal glycemia in an aged mouse were somewhere outside the beta cell. What would you predict would happen to insulin secretion? Well, the beta cell, in its role as a nutrient sensor for the organism, would start to relax. It would release less insulin because there's less insulin demand. But that's not what happens. The mice that maintain glucose tolerance on a Western diet are releasing more insulin, not less. So, like I said, I can't prove this, but I think it's a really interesting theory, and maybe we could get some experiments to test this. So that brings me to my acknowledgments, and the first one is for my wife. We have a new baby. So this is our daughter, Ryan Elizabeth, with our son, Brendan. And these two were a handful this week, and I really want to thank my wife because everybody got sick earlier this week. Brendan had a fever, I had a fever, and my wife took care of everything so that I could stay up and make this talk. So my heart goes out to her. But I want to talk about where all of this work was done. So in my lab, we don't sort of have this view of lone wolf science. That's really not how science is done anymore. We do this as a team. So despite the fact that Trilly Gregg was the first author on most of this, there were aspects of data in here from every single person in my lab. I also want to thank our undergraduates, our funding sources who were funded by the NIA, the NIDDK, and from Wisconsin, from startup funds. And it's really the NIA award that was foundational for producing these studies. And so the NIA allows us to obtain these aged mice where we can very quickly phenotype them without waiting 2 years. And these studies were all done in collaboration with Dudley Lamming's group. And I especially want to thank Dudley. He's been a great friend. And the people in his lab have been wonderful. And this is the list of collaborators that's on these publications alone. This is not an exhaustive list. So it takes a large group to actually go from an organismal phenotype down to a single molecule. These studies are quite simply not easy to do. So it takes a lot of people to do it. Thanks to everyone in the VA4C labs and to Jeremy Smith, who graciously watched me give this presentation on Monday so he could give me some tips. Thank you, guys.
Thank you very much, Matt, really for an outstanding talk. And it harkens back to the first time we met, and I asked you about your science, and you so clearly were able to explain what you did, and it only increased my enthusiasm for Vince's recruiting you. Also to be part of a group which has come to fruition, the group that you're acknowledging here. It's really a wonderful thing to see, and also I think you all are leaders in the world, really, in beta cell function and science. So congratulations to you and Vince. I'll ask you to call on the audience, and please repeat the questions.
Oh, sorry, wait, Robin first. I'll get to you in 1 sec.
Have you been able to show this exercise to reverse any of these changes?
You can repeat the question.
The question is, has exercise reversed any of these changes? So we've not tested it. Exercise basically helps everything. The The best genetic evidence I have for you is Steve Britton and Lauren Koch at the University of Michigan bred rats that run really well together and rats that run just a fraction of the day together. And they kept breeding these populations apart and apart and apart. And that breeding strategy where they've got these high-capacity runners and these low-capacity runners has rescued every defect that they've ever thrown at it in their long career. So I suspect that exercise would impact this. I really do. But what we would need to do is we would need to age out these mice and then run an exercise study, and we just haven't done it. But it's a really interesting question. I mean, age and calorie restriction, there are hints in there about therapeutic targets that we haven't even tapped yet. Lots of researchers are trying, but we're just not quite there yet. Robin, you had a question?
When you look at the effect of age on the ATP-sensitive potassium channel, can you tell over time, is that channel degraded, there but degraded? Is it relocalized? Is it there but not functional?
Sure.
Or is there something else going on?
So Robin asked, given the changes in ATP-sensitive potassium channels in the plasma membrane with age, what is it about the channel? Is the channel degraded? Is it relocalized? Yeah, we think it's relocalized. So in order to get a channel to the plasma membrane, you think about its history. So the protein has to be synthesized. It has to go into the ER and Golgi, and then it has to be shipped out in a secretory vesicle, which then fuses with the outer membrane of the cell. Right? That's the process. So under certain physiologic insults like endoplasmic reticulum stress, this is an organellar stress that's a major risk factor for type 2 diabetes. Those channels get stuck in the endoplasmic reticulum. There are also physiologic mechanisms that adjust the trafficking of the ATP-sensitive potassium channel in and out of the plasma membrane. One of them has to do with cell senescence. So it's recently been shown in a high-profile paper in, I think, Nature Medicine, that some of the senescence factors that affect the cell cycle affect cyclin-dependent kinases. There's one cyclin-dependent kinase inhibitor that's famous in the aging field. And the cyclin-dependent kinases themselves, we found, our lab has found, can adjust the number of KATP channels in the plasma membrane. And my student Sophie has knocked out that cyclin-dependent kinase in mice to test that specific hypothesis. And she's going to be actually doing those experiments within a month. So good question. Vince?
First a comment.
Yeah.
Well, you are really smart. And it's really remarkable what you've done in just a little over 2 years, because I remember our divisional administrator going to Home Depot to build benches in the apron. So
It's true.
happened in just 2 years. So then the question then. So would it be cool if you could prospectively identify the mice that go into the NGT versus IGTs? Are you guys thinking about that at all?
Yeah, so Vince's question is, so what if we could prospectively predict which mice would get diabetes from which mice would not? Which would compensate for obesity, which were not? So the answer to that is coming soon. So I have a colleague at Indiana University, Carmela Evans Molina, and she is part of a group of diabetes researchers there, and they've just gotten a DRTC, a Diabetes Research Training Center, which gives them essentially an enormous pile of money. What they're going to do with this money is they're going to start imaging the pancreas in living mice. Now here is where I think the lifetime technology that I talked about can come into play. What researchers at Wisconsin have done, researchers like Roz Anderson, have done is they've said, all right, well, let's take the lifetime imaging signature and let's hit these cells, let's measure these cells, and let's predict which of them will respond to treatment. And that's in a culture dish. And of course, what I'm talking about is actually doing that in the living animal, and that's what your question is focused on. So, yes, I think we're probably going to need another 3 or 4 years before we can get it sort of a tractable approach down on that. As you know, it involves quite a bit of cutting into the mouse to actually get to the pancreas. It's a really hard organ to image. But yeah.
Okay. With
Thanks.
that, I want to thank Dr. Merrins for an outstanding grand rounds.